exciting day for us and a very exciting opportunity, and I'm very honored to be able to kick things off today. So I would like to say hello and welcome to each of you who's joined us for this time. Just to give a little bit of an introduction before we go ahead with our exciting lineup, I would just like to talk about where we are and what we are doing here today. And to start that off, I'd just like to think about the COVID-19 pandemic, which we've spoken about and heard about a lot. And it felt like a problem of the developed world until March the 5th, when South Africa registered its first index case. Since then, life in South Africa has followed the same roller coaster ride experienced in most of the world's 212 countries that have experienced COVID-19 cases. We've been in a state of national disaster since March the 15th and under lockdown since March the 26th. And now we're in a position where we're waiting anxiously for the relaxation of the lockdown levels. Our optimistic selves had hoped that, first of all, Africa and South Africa in particular would have miraculously been spared this nightmare. Then, our optimistic selves hoped that against all odds, the curve would be flattened into disappearance. Unfortunately now, by day 55 of the lockdown, reality has settled in for most of us. COVID-19 will most probably be our global companion for the next couple of years. We desperately, urgently need to find a very broad-based new normal, especially when it relates to clinical research and drug development. It's time to face reality, to jump into action and prepare to continue with our essential work, because patients suffering from COVID-19 and non-COVID-19 cases need us. This is what today is all about, starting the dialogue. To do this in a far superior way to what I would manage, we have the pleasure of hosting Professor Abdool Karim on our webinar today. Before introducing the professor, however, I'd just like to cover a few other elements. And the first is why today? 20th of May, as I think a number of you know, marks the day of International Clinical Trials Day, a day familiar to everyone working in the industry. But since COVID-19, our industry has gained far more interest, and this is a day now important to so many more people. Before we get started, I just want to cover a couple of housekeeping points. As you'll have noticed, due to the large number of attendees, your lines have been muted. They will have to stay this way, unfortunately. Please will you utilize the chat panel to ask any questions that you would like to. Throughout the session, these questions will be compiled, and a time-dependent number of them will be asked of Professor following his session. The meeting recording, as well as the responses to the compiled questions, will be made available on demand to attendees following the meeting. I would like to take a moment to thank BioCare for sponsoring today's event. Leonard Lazarus from BioCare will spend a little time talking to us later, outlining what they do. And as a result, I'm not going to go into it now, but I do want to extend SACRA's gratitude to BioCare for the contribution and support they show SACRA, both today and on an ongoing basis. Thank you so much, Len, and thank you so much, BioCare. Right. Professor Salim Abdool Karim is a South African clinical infectious disease epidemiologist. He's ranked among the world's most cited scientists by the Web of Science and widely recognized for his scientific contributions to HIV prevention and treatment. He is the current director for the Center of AIDS Program of Research in South Africa, more widely known as CAPRISA, and he's CAPRISA Professor for Global Health and Epidemiology at Columbia University. He is currently serving as the chair of the South African Ministerial Advisory Committee on COVID-19. Ladies and gentlemen, I would like to now hand over to Professor Salim S. Abdool Karim. Thank you, Professor.
Okay, great. Thanks very much. Really, it's a great pleasure and an honor for me to be here with you this morning. What I hope to do in the next 25 minutes or so is to share with you some information on the COVID-19 epidemic, look at where we are in terms of the trends in the epidemic. I'll spend a little bit on the Western Cape because there are some important lessons to be learned from there. And then I'll end off with just making some comments about non-COVID clinical trials. Actually, they apply just as well to COVID clinical trials before making some conclusions. Okay, so what are we trying to achieve in the South African national COVID-19 response? And this has been some source of confusion, because some people saying that we're trying to end the epidemic, or some people trying to think that we are getting to a point where, you know, the epidemic is just going to go uncontrolled. So our goal, the national COVID-19 response goal, is simply to flatten the curve. Now, flattening the curve, the purpose behind it is to ensure that not ensure, but to aim to get cases and the clinical burden to a point, and to maintain it at a point where the health service capacity exists to care for patients. Now, why would we want to do that? We want to do that because of what we saw in Italy, what we saw in New York, that if left unchecked, the way in which this virus spreads is so rapid that there is a very quick accumulation of cases, and it begins to overwhelm the healthcare system. So why are we trying to flatten the curve? What are we trying to achieve in this process? Well, without natural immunity, and right now we don't think anyone has natural immunity if they've not been infected by this virus, or a vaccine, everyone is at risk of infection. That means that our entire population of 50 somewhat million are at risk of infection. Doesn't matter whether you're rich or poor, green, blue, or men, women, doesn't matter. Every one of us is at risk of becoming infected. And we know that about 1 in 5 of the sick individuals who have symptoms may need hospital care. And that's based on the evidence that came out of China from their first case series of about 45,000 patients. So flattening the curve is an ongoing process. And I've heard some people say, oh, we've flattened the curve, or you haven't flattened the curve. Well, I'm sorry to tell you that it's not a once-off event. It is something that we are going to be doing for a long time. And that what we've just achieved at this point is we've just started flattening the curve. And that we have to expect that we will be doing this for quite an extended period in order to reduce coronavirus spread. The reason for flattening the curve I already touched, which is to reduce the rate of new infections so that the peak is lowered to a level where hospitals can cope. And that reducing the peak of the massive surge that we're going to see of very sick COVID-19 patients is that we want to avoid overwhelming the healthcare provision, because hospitals that are coping provide better care and lead to fewer deaths. The last thing we want is to see what happened in New York, where patients got to the hospital and they couldn't get in because the hospital was totally full, and patients were, struggling to get medical care. So what flattening the curve is not. It's one thing to know what it is. Let's look at what is it not. So it's not a single event. It's not about eliminating new cases. We're not sure if anybody can do that. Certainly, we've seen very successful efforts at flattening the curve, for example, in New Zealand, but they're not out of the woods. The moment they release and ease their restrictions and people start moving, they run the risk of getting a second wave. So it's very hard to get to a point where you've eliminated new cases, and probably at this stage, close to impossible. And so we're not trying to do that, and flattening the curve is not about that. It's not about stopping the epidemic. It's not about eliminating outbreaks that will occur. And it certainly is not about avoiding any deaths. So deaths will continue to occur. So now I've given you some idea of what flattening the curve is and what it isn't. So why did we want to flatten the curve? And why did we want to do so early? And we are among the few countries that was able to do it early. New Zealand being another example, Australia being another example. Those countries that were later in the epidemic, when their first cases occurred, were better prepared and could act early. Well, it turns out that early seems to be better. Flattening the curve in advanced epidemics has turned out to be very difficult to achieve, as we saw in the UK, when they were already very far advanced and tried to institute a lockdown in the hope that they would flatten the curve. Well, the cases were just coming at rapid fire to the medical services. So early seems to be better in terms of having an impact. The four reasons that we implement the lockdown is firstly to slow community transmission. So we know that the first cases occurred in travelers. We know that their contacts became infected to some extent, but the transition in establishing widespread community transmission, that has not taken hold in South Africa in the way that we had anticipated. That we had expected it would rapidly establish itself in the community and would spread. Well, for the most of the country, that has not occurred. And that is no reason for complacency, because that's just what the situation is now. Tomorrow could be very different. But it was to slow community transmission. The second was to provide time to expand our healthcare capacity, especially ICU and high-level care. The third was to need the time to better prepare and equip hospitals and healthcare workers. And then the fifth was to provide time to scale up our testing and our prevention strategies. So those are the four reasons why we needed to institute a lockdown. And the earlier we do it, we thought may be better. So what has happened in the epidemic? Well, let's start with tests. When you look at the number of tests, you see that initially we were doing very few tests, as you would expect, because there were very few clinical patients coming in. We had criteria relating to clinical conditions, and so we did few tests. We should have done more tests as time went on, but we were a bit slow in trying to ramp up testing. But when we did ramp up testing, we did so in a very dramatic way. Not dramatic, but in a way in which we saw regular increases week by week in the daily number of tests that were being undertaken. And you get some idea here as to the number of tests, and then I've shown you separately how many of those tests were being done in the public sector. So there's no question we have ramped up testing. Let's look at the actual situation. So the number of tests is in orange, and the y-axis for the number of tests is on the right-hand side. We are almost at 500,000 tests that we've undertaken. In terms of cases, the cumulative number of cases is in the blue line with the blue dots, and the active cases are in the blue bars. And it's more appropriate to look at the active cases, because cumulative cases are not totally informative because of people who've recovered or who've died. So the active cases give you some idea of what our epidemic curve looks like. What about doubling time? Well, doubling time is just one indicator that's used to get some idea of how the epidemic is growing. Now you have to know that doubling time is dependent on tests, it's dependent on lots of things. And so you can't just treat it in a completely uncritical way. It has lots of limitations. But before the lockdown, the epidemic was doubling roughly every 2 days. And at a very early stage in the epidemic, you would expect that the epidemic would grow fast, and once it's established, it would start slowing. But we had a very rapid growth initially in the first sort of 3 weeks or so of the epidemic. And during the lockdown, we had a slowing down in the rate at which it was doubling. We went from 2 days to 15 days. And since the easing of the lockdown, we've maintained a pretty reasonable rate at which the doubling time has come down. It's now in the region of around 12 days. So that just gives you some idea of the rate of growth of the epidemic. Again, take it with all of the caveats that I've mentioned. So one of the most important indicators or markers that help us understand an epidemic is something called the reproduction number. And that, put very simply, is just the number of new infections from each infected individual. So there's a person here with infection. How many people is that person going to infect? And that reproduction number, referred to as R0, is a very important measure of how the epidemic is growing. Now, the problem with R0 is very difficult to calculate. And usually we calculate it once we've got quite far down the epidemic because it needs a lot of data. So instead, people have developed another number called the effective reproduction number. And the effective reproduction number gives you some idea of how the reproduction is changing. In other words, how the R0, it's a sort of indicator of R0. It's not a measure of R0. You can't measure R0 in this way, but it gives you some idea of what R0 might be. So call it a rough estimate, if you like. And it's based on taking the cases over the last 7 days and then predicting based on Bayesian theory what the next number of cases is going to be. And you can just shift it a bit so that you get to, instead of looking at cases, you look at infections. And that's what I've done here. And when you look at the effective reproduction number, you'll see that when the epidemic first started, we had a pretty high reproduction number. And that was evident again in the doubling time. That for every case we had, the number of new cases were quite substantial. But you can see that in the gray zone is the confidence interval. So there was a very wide confidence interval initially because you're dealing with very small numbers. And then with the onset of the lockdown, the state of emergency, the number of cases went down, and so you saw the effective reproduction number go down quite substantially. And then while that effective reproduction number was at this low level and was starting to rise back up, we instituted the lockdown. And in the course of this last several weeks, the reproduction number has been, don't worry too much about the up and down because that's just reflecting the rate at which cases are reported. There's a lot of variability in that. But essentially, we've been sort of hovering around the 1.5 or so. Now, what that means is that if the reproduction number is above 1, it means the epidemic is continuing to grow. In other words, each infected person is infecting 1.5 new people. Or you can look at it slightly differently. For every 2 people with the infection, they infect 3 others. But it's not 2 or 3. In other words, it's not the reproduction number. Let's say a reproduction number of 3. If you see a reproduction number of 3, that means each person with this virus is infecting 3 others. And that is probably closer to where the real reproduction number for this virus is, somewhere between 2 and 3. But because we've instituted control measures like social distancing and hand washing and so on, we don't have such a high reproduction number in South Africa. Our reproduction number is closer to 1.5 or so. So we're not seeing that exponential increase in the number of cases. So what's happening in the hotspots in the Western Cape? So when you look at the epidemic in South Africa, what's very clear is that it's quite different in different parts of the country, in that in some places it's growing rapidly, in others it seems to be sort of meandering along. And if you look at the four biggest provinces, the ones that have the highest numbers, again, you see that the cases are particularly rising in the Western Cape, but not to the same extent in the other three provinces. And if you look at doubling time across these different provinces, again, remember, doubling time is dependent on testing, it is dependent on cases, dependent on lots of other things as well. But it just gives you an indication of how fast the epidemic is growing. And you can see the differences that we have in the doubling time across the different provinces. So South Africa doesn't have one epidemic. We've actually got a mosaic of epidemics at different stages in different parts of the country. If you look at testing, well, testing has been progressively going up across pretty much all of the provinces, and Gauteng, leads the way with testing. They have a very high number of tests. Roughly 1 out of every 3 tests being done in South Africa is being done in Gauteng. And the overall positivity rate remains low. And that doesn't matter whether you look at hospitals or whether you look at the community, or whether you look at the private sector or public sector, it's pretty much low all across. Whereas in the Western Cape, that accounts for about 1 in 5 tests that we do. You can see that the positivity proportion is higher at about 8.4%. And so the number of cases is high, as you've been seeing. It has the line that's going up. This probably gives you a better impression of that. And this is the positive test proportion. So if you look at the positive test proportion, that's the number of tests that are positive of all the tests that are done. And this is a better indicator because it gets rid of the problem of cases occurring as a result of testing. This is a fraction of the test. This also has its limitations, and one needs to be careful in interpreting it in an uncritical way. But if you look at the green bars tell you the proportion of tests that are positive in all of the provinces besides the Western Cape. So the green bars, if you look at, say, for example, the week of the 17th of April, there were 21,682 tests done, and of those tests, about 2.3% were positive. By the way, that basically means that we are doing just over 40 tests to get 1 positive. So that just gives you some idea of how uncommon the disease is when you have a positivity rate of 2.3%. So it's not a problem that you're not testing enough, you don't have enough patients who are positive among those you are testing. And if you look at the next week, the week of the 24th of April, you can see they increased the test by another 10,000. So we did 31,000 tests in all the other provinces, and we have 1.7%. And then we increased it by another 10,000 the week of the 1st of May, and you can see it's 1.8%. So in the rest of this country, the test positivity rate has been fairly low. And that just sort of, even though testing has increased, and so the number of cases is going up, but the proportion that are positive has remained within a narrow band. Now that is not true for the Western Cape. In the Western Cape, in the gray bars, you can see that in the week of the 17th of April, they had about 8,000 tests, and they were about 5.5% that were positive. In the week of the 24th of April, they had 10,000 tests or so, and 7.9%, and so it goes on. That as we increase the number of tests in the Western Cape, it's not just that we're increasing the tests, then the proportion that are positive is going up, and that's going up in our hospitals and so on. And that's become very obvious when you go to the Western Cape. number of patients in the ICU, in the hospitals is much higher. You don't see that when you go to the other provinces. So what's going on there? Why is the Western Cape got this different curve from the rest of the country? Well, essentially, it's about hotspots. And when we look at the way in which the South African epidemic occurred in the bars, the blue bars, that's the number of active cases, we have an 8-stage strategy of how to respond. Our response at a national level comprises these 8 stages. So we finished the first 4. We finished stage 1, the preparation. We finished stage 2, primary prevention. We finished at least the stage, the phase, alert level 5 lockdown, and we are now in alert level 4 and going down to 3. So we'll ease off the lockdown over the next period. We've finished the stage 4, which was our active case finding, where we have thousands of field workers going house to house to do active case, looking for patients. And we are now switching to hotspots. And in the Western Cape, they started working on hotspots about 2 weeks ago. In the rest of the country, they're slowly building that capacity and initiating the hotspot part of our response. What's involved in hotspots? Well, the first is to identify the hotspots. Where are they? And how do we know where they are? And so that involves some basic epidemiology. We need to know where the cases are occurring. And then we need to intervene. We need to understand the spatial nature of the new cases. We need to do outbreak investigations. And we need to understand why the virus is spreading in these little outbreaks and hotspots. So what kinds of hotspots are we seeing? Well, I'm just showing you some graphs that come from our colleagues in the Western Cape. In here, in the Western Cape, they've been trying to identify their hotspots, and they have used this kind of spatial technology to do so. So if you look at the top picture, Khayelitsha, you can see Khayelitsha has quite a well-defined hotspot. And a lot of it revolves around the Khayelitsha supermarket. And it's turning out in the Western Cape, supermarkets, in fact in other parts of the country as well, supermarkets have tended to be quite important sources of hotspots. You can just think about it for a moment, that an infected individual who gets into a supermarket contaminates the surfaces. The staff then pick it up, and then they get infected, and they spread it to other staff, and then they contaminate surfaces, and they pass it on to other customers in the supermarket. So you would expect that there will be these hotspots, and the same with Delft and in factories like the GSK factory in the Western Cape. So these are they have some idea of what their hotspots are, and they have some idea of where the virus is spreading. The challenge becomes that the hotspots quite often become diffuse after a while, because if you take like a supermarket, the customers come from a wide range of places. So now you have actually the virus seeded in quite a wide spread. So it becomes general community transmission at that point, and it's very difficult to control at that stage. You have some options when it's still a hotspot, but once it goes beyond a hotspot and becomes diffuse, it's a bigger challenge to contain. And remember that this is not a cause for concern, because that's how this virus spreads. So this is not unexpected. We already knew we were going to see these hotspots. We even had a whole stage of our response ready, and not ready, but preparing for this stage of our response. And in the Western Cape, they've got a whole hotspot strategy in which they're dealing with hotspots. So I think I've given you a sense of what the trends are in the epidemic. I've given you a sense of what's unusual in the Western Cape. And I think pretty much what we're seeing in the Western Cape, we'll probably see in the rest of the country, probably in about 3 to 4 weeks later. Roughly, my estimation is around that sort of time frame. Now, don't hold me to it, but that's a rough sort of guess. So what's happening? How are we doing in relation to the rest of the world? Well, I've used this diagram before to make the point that the South African epidemic tracked with the UK epidemic initially. And this is at a point where both our countries had 100 cases. And from the 100th case, if you plot the two, our trajectories were pretty similar. The UK, which did very little by way of implementing interventions because of the political decision-making in the UK, they're an epidemic that pretty much grew very substantially and exponentially. And on the other hand, our epidemic didn't. If you look at our epidemic, well, we sort of, dare I say it, we've flattened the curve, or we've started flattening the curve. And in a way, that's a paradox that we deal with today, that because we acted quickly and we started flattening the curve, we actually don't have much of a problem. So the hospitals and the clinics and so on are pretty quiet, except in the Western Cape. And so everybody's asking the question, so why are we sitting at home if there's no problem? Well, we don't have a problem because we're sitting at home. So it's a chicken and egg problem that we're dealing with. And so people need to understand that this is achieved because of the interventions that we implemented. Now, we don't know any of this for sure. Now, if people say, okay, we've definitely done this and we definitely didn't do that, you can only say that if you have a counterfactual. In other words, I mean, this is the last group I should talk to about counterfactuals, right? Because you do clinical trials. You would like to know what happens if you don't intervene. And that's why we have a placebo control group. In this particular instance, I don't have a placebo control group. I just have two countries. And so these two countries are not even comparable, right, by many indicators. But I use it as a way to illustrate that we could have gone the way of the UK and we didn't. Right? And to the extent I believe that it's somewhat a function of what our interventions were, you can take it or leave it. But I would say that they've contributed in some way. How are we doing in terms of the rest of the world? Just to give you some other countries. So New Zealand, Vietnam, Australia, South Korea, all examples of very successful efforts to control the epidemic. Actually, Singapore also was. But as we're seeing, in the case of South Korea and Singapore right now, they're all back to square one. They've all now got growing epidemics. It's in the nature of this, because even if you flatten the curve, the moment you lift the restrictions, you have people moving, and then the virus spreads again. So you can't win here. You flatten the curve, and then you release it. You release your interventions, and things go back, and they start increasing. So South Korea has got a pretty rapid increase in cases. Singapore has had outbreaks in a big way in some of their hostels. But we're sort of muddling along, and our country is sort of, at least at this point, in the pack of countries that are doing reasonably well. Given that I think we are now around day 50, day 52, after the 100th case. We use the 100th case because until you get to your 100th case, there's a lot of variability, a lot of background noise. So taking from the 100th case onward, you can see that there's a lot more stability, and you can look at the data and interpret it more easily. How do we compare on the 52nd day, which was on the 14th of May? Well, you get some idea of the situation. Remember that our population and the UK population is similar in terms of number of people. Of course, the nature of the movement and the nature of the GDP and so on is very different. But if you just look at the UK, they on the 52nd day had about 161,000 cases compared to South Africa, where we had close just about 10,500 cases. So at similar points in the two epidemics, we are at around 10,000 cases in league with South Korea, whereas the UK is more in line with what we're seeing. Okay, let's go on to clinical trials. So we all understand that this virus is going to be with us for a long time. This is not something that's here today, gone tomorrow. As I just explained, in South Korea, they now are in the midst of their second wave in the epidemic. So we're going to have to figure out how we get on with our lives and get on with doing what we have to do. And the way in which we do that is we use our prevention toolbox. And in our prevention toolbox, these are all the tools, and it'll grow over time, but this is what we have now. And we need to be able to pick from this toolbox the most appropriate set of tools that we need to solve each problem. So in a particular setting, if we go, let's for example say, if we go to a school, these are the tools we'll pick out from the toolbox. If we go to a workplace, we'll have a different set of tools, and so on. So we need to find which combinations of tools work for us. Now we need to be careful that, let's for example say that if we are trying to knock a nail into a piece of wood, that we don't try and do that with a screwdriver or with a spanner, that we do that with a hammer. So we need to make sure that we pick the appropriate set of tools to fix each problem as it arises. And so it's easier to think about it as a prevention toolbox that we'll pick our tools from, and we'll use them in differing combinations. And at this stage of our response, as we focus on our hotspot, we'll pick our tools to deal with each situation. Because we can't sit back and say, okay, the virus is going to be here, we can't do anything, we're going to sit at home. That's not a sustainable future, because we're going to be staying at home for a long time. We don't anticipate we'll have a vaccine anytime soon. And remember, we may end up never having a vaccine. So how do we get on with doing what we need to do? So we've seen that the level 5 and level 4 lockdown has had quite a few negative effects, especially on aspects of healthcare, in terms of, we're not picking up enough TB cases, we've got patients defaulting with their HIV treatment, they're delaying visits, and we can't even do our clinical trials. And those are important because we've got to find solutions to the HIV and TB and all the other problems we have in the midst of COVID. We can't say we'll put our life on hold for COVID, and then when COVID's over, we'll then come back and pick things up and carry on. That's not really a possibility or a pragmatic way to think about the way forward. So how do we go forward? Well, we need to continue our clinical trials as safely as we can. And we can do that through a range of things that we need to do. The first is to put forward to ethics committees that we have a very clear strategy in which to continue doing clinical trials, and we can do so in a manner that's safe enough. It'll never be totally safe because we will expect to see outbreaks occurring, and they will occur even in research settings. But we need to try and minimize those, and we have to have very clear and rational plans picking from the toolbox in order to be able to say that coming to a clinical trial setting and participating in a clinical trial doesn't pose an inordinately high risk of transmission of COVID. And that one of the key things in doing all of this is to ensure that our clinics are well organized and well designed to be able to screen and triage even before anybody enters a clinical setting. And now that's being implemented in every hospital, every clinic. That when you enter, you need to ensure that you are being screened and that you are then triaged to red or green zone. Red being patient that's either proven COVID positive or COVID suspect, what used to be called previously a PUI, person under investigation, or somebody who was coming in for non-COVID related activities. And then that person can be seen in the green zone in our research clinics. So let me conclude by making some points. The first is, I think that we've at least averted the exponential growth in our epidemic curve. And that's, we could have gone the way of the UK and we didn't. We've started flattening the curve successfully, but we're going to need to continue doing that for quite a long time. We've had time to improve our testing, and you've seen how that has increased. We're building a lot of new healthcare capacity. We're building field hospitals. For those of you who haven't seen the Cape Town ICC, if you have a chance, it's quite amazing. I've just seen the first set of pictures from there. All the partitions are in, they're bringing in the beds. So we will have a major new field hospital in the ICC in Cape Town. And in fact, in all our major centers, we are building these field hospitals. We are preparing for that onslaught or surge of cases so that we have enough clinical capability. Our testing coverage is improving, but we've got problems, and those problems are around supply chain. Given the challenges right now with aircraft and so on, our community transmission has mostly remained low and is not increasing exponentially. The Western Cape is an exception, and there in the Western Cape we are seeing multiple outbreaks, and those multiple outbreaks are now seeding into general community transmission in some districts. The effective reproduction number is still hovering around between 1 and 2 in the Western Cape, and Eastern Cape somewhat, but it's closer to 1 in the rest of South Africa. It's not 1, it is above 1, and so the epidemic is growing in the rest of the country. It's just not growing exponentially. All of this tells us that there's simply no room for complacency. The case numbers are going to rise. We have to expect outbreaks. And if you watch my presentation on Easter Monday, you'll see I talked about the flames. We're going to see these flames everywhere. They're going to occur. They're going to occur in our shopping centers. They're going to occur in our parks. They're going to occur in the way we work. And we shouldn't get concerned about that. We're expecting that. That's how the virus spreads. So we've got to identify these outbreaks. We've got to mitigate them. We've got to carry on doing what we need to do. Otherwise, we'll pretty much close everything because we're going to see these outbreaks. They are going to occur. And what is important is that every time we see these flames, we try and prevent a raging fire. And so that's why when we look at the Western Cape, it's giving us an indication of what we're likely to see in the rest of the country. It's sort of a forerunner of what may happen elsewhere. And so we need to be very clear that we focus right now on stage 5, the hotspot identification intervention, especially in those parts of the country where we are seeing these hotspots, because we've got to be there, see those flames, identify them with vigilance that we need to put in place, and that we prevent those flames from becoming raging fires. I'd like to thank many of the people that have contributed to putting this information together. I hope I've given you a good sense of the challenges that we deal with. I'm going to now try to undo my sharing, but I'm not sure if I know how to do that. There we are.
Hi, Professor. Nikki Stott here again. Thank you for that very engaging and enlightening presentation. I'm just going to share my desktop. You're welcome to unshare your screen, but if you wouldn't mind keeping your video up so that we can take this opportunity to ask you a few questions related to the content you've shared and some general questions that have shown through the chat panel during your presentation that relate to the situation as a whole.
I'm trying to figure out.
You're good. I can see you, but I can't see your screen anymore. So I think that's good.
Okay. I just wanted to try and get rid of the slides, so now you can see me. Okay, you can do that.
I can see. Okay. Thank you, Professor. Thank you once again for that presentation. Now, I've gathered a few questions from the chat, and I would just like to find out from you, is the hotspot information more broadly accessible for the general public, just to know what areas to avoid and which areas are currently safer?
So the hotspot information at this point is being developed at a provincial level. Mostly the hotspots we're seeing are in certain sub-districts and districts. The minister about 2 weeks ago provided a map of South Africa where he identified, the districts which have the hotspots. The exact location of individual hotspots is not very easy to do because it needs a level of precision in getting addresses and so on that's not always available. And that's being dealt with more at a district level where they're trying to identify very narrow hotspots. But the information about which districts are hotspot districts, that's already known and was presented.
Thank you. Yes, so that answers my question as well, because I was wondering that spatial technology you showed for the Western Cape areas, was that done for all the hotspots across the country?
So the hotspots I showed you for the Western Cape, that is the technology they're using in the Western Cape. In Gauteng, they use a different technology.
Okay.
And we have a national platform actually based at the CSIR. So in the Central Operations Center, there's a big room, a hall, with the whole wall full of TV screens. And there is a big map of the country there that has all this information. But there are many challenges to the data right now. I describe it probably simplest as, we're sort of sailing the ship while we're building it. And so we can sink at any time because we're still building the ship. And so a lot of the technology we're using, they don't talk to each other. And so there's many challenges in getting all of that up and running. But that's the idea, that at some point the ops center, you can walk in there and you can just see it and we can share that. But it's not ready yet. All of this takes more time than the epidemic gives us.
Sure, and I like your analogy to the sailing ship, seeing as we're celebrating Clinical Trials Day and the first documented clinical trial was the one on the ship. So, so thanks for continuing with that theme. Okay, let me move on to my next question, and that's with the comparison that you did with the UK. This was in their winter, which is ahead of our winter. So will this information and will this trajectory hold true for the next 6 to 8 weeks?
I don't know. I often say, I wish I had a crystal ball on my desk. I could sort of look into it and tell you what's going to happen. Sorry. I just have the same information all of you have, and I try to analyze it as best as I can. And you can ask the mathematical modelers. I'm not a mathematical modeler, so they have done many projections and so on, and they can give you better information on these things. From my way of looking at it, the data pretty much is as I presented it, and it gives you some idea of what you can see in the next few weeks. If you go beyond that, your guess is as good as probably anyone else's.
Thank you, Professor. Another question, excuse me. It's extremely important that we continue with the development of generic products to drive affordable healthcare. And do you foresee any issues with the conduct of clinical trials in healthy volunteers during the COVID-19 pandemic if sites have appropriate procedures in place? And you have touched on this a little bit, so just to put it in a different context, if you wouldn't mind.
I think I was just pointing out that we're going to have to live with this virus for quite a while, several months, if not years. So we have to carry on. We have to do all the things we have to do. And we have to do so in a way that minimizes and mitigates the risks associated with it. And if we can do that, there's no reason why we shouldn't get on with things. There are some things that are very hard to mitigate risk. I mean, to me, going to a sports stadium, that's a difficult one to mitigate, because you have an exposure in that kind of setting, and you've exposed thousands, and it can just rapidly get out of control. So there are certain things that are very hard to mitigate right now, and we just need to understand that those are difficult things to mitigate. But there are many things where we can mitigate this, and clinical settings is one of those areas where we've already learned how to mitigate risk. We know how to do that. It's not rocket science. There's a few basic elements that you put into place. If you do those things, you can mitigate risk. You don't avoid risk. In other words, it's never down to zero, but you mitigated it to the point where it's practically a way in which you can continue to function. So to me, that's how we should think. That's how we need to think, that when we get to level 3 and we can begin to get on with things in a much more open way, taking our mitigation factors and our toolbox into account, we should do that. Now, it has to be said that as we do that, we have to understand that the cases are increasing, and they're going to continue to increase. I'm not telling you that you're going to go into a future that, okay, everything is going to be safe and so on. The cases are going to go up. We are going to see outbreaks. Let's take that as a given. How we deal with it and how we live with it, how we manage it, is how we define the way in which we move forward. But there is going to come a time when the surge starts, and the surge may start differently in different parts of the country. When that happens, we may have to, again, reduce our interactions and so on. But we'll deal with that when we get to that point. Right now, it's not in the immediate horizon, perhaps except in the Western Cape, but for the rest of the country, that's not applicable at this point.
Thank you. And I think opening the schools is also something that people are wondering where that fits into the analogy around the visiting the stadium versus the clinical setting.
Yeah.
So I think that will be an interesting scenario as well.
I think one of the most interesting things that we have seen, that's quite striking. Abdool in last week's New Yorker outlined this. Abdool is the very famous surgeon from Boston who wrote the book called Checklist. And he pointed out that actually in healthcare, we have figured out how to mitigate the risk, in that we have very few cases of COVID in the COVID wards. It's quite striking, actually. When we did the investigation here at the team did it from UKZN, the investigation in St. Augustine's Hospital, there were lots of staff and patients that became infected with the coronavirus, but not one from the COVID ward. And the staff in the COVID ward, none of them became infected to date. So if you are careful, and it's not just a PPE issue, it's about a mental state. It's about knowing what your risks are and being conscientious about implementing your toolbox. So I think if you do that, we can see how we can mitigate this risk.
Absolutely. Thank you, Professor. We have a few more minutes left, so I'm just going to have a look at a couple more questions. And one here that we've received from the chat, the latest modelling for South Africa suggests 40 to 45,000 deaths by November 2020 and positive cases in the region of 1 to 1.2 million by November. can the healthcare system cope with these projections?
As I explained, I'm not a mathematical modeler, but I've seen these models, and they look quite reasonable to me. And I'm not sure how well we would cope with the entire surge. But I think to the extent we flatten the curve, to me, is more important. That our goal must be not to have 1 to 1.2 million positive cases. Our goal must be to have fewer. And if we're going to have 1 to 1.2 million cases, that's okay. We should just have it over a longer period so that the cases, when they come into the hospitals, are at a level we can cope. It's hard for me to say right now whether we're going to be able to cope or not, because frankly, we have no idea how much we have blunted the surge. So have we really avoided that big peak? I hope so. Do I have evidence for that? No, I don't. We are largely in a situation where the data are limited, and we're just learning from what we can from other countries, knowing that other countries are very different from us. I mean, the classic example is, most of the really good studies in schools and children come from China, Europe, and the US. Those schools are very different from our schools. So you can't just wholesale take that data and apply it in our schools. We have to understand the limitations and what our conditions are differently. So we try to do that with all other countries. We look at what other countries have done. We try to see how much of it is applicable, what of it can we borrow, what might work, what might not work. And then we try to create some extrapolations about, okay, this is probably one of the better options going forward. But I can't tell you whether we succeeded or not, whether we will not have a surge that we can't manage. It's beyond any of us to tell that right now. I can say that if we did have our surge, like the UK, we would have been in the situation that was in the UK, which is an uncontrollable, unmanageable situation with large numbers of deaths. I hope that that won't be the case right now.
Absolutely. Thank you. I would like to just direct the questions to a little more clinical research questions. And the first one, could you tell us about South African or African innovative research, as opposed to, are we only doing Western-driven investigations into COVID-19?
I'm not the best person to answer that. As far as I'm aware, there are something like about 5 or 6 clinical trials in South Africa underway right now. One is of a BCG vaccine in healthcare workers. There are 1 or 2 trials of chloroquine or hydroxychloroquine. And there's the big WHO Solidarity trial. That is a 5-arm trial that's being done in many countries, including South Africa. So those are among the trials I am aware of. I know that discussions are underway by several groups to look at participating in yet more international clinical trials. I have not, at this stage, seen trials that are purely South African and are completely new ideas. I guess closest it comes to is the BCG trial, but that's not new. Actually, that's being done in Australia. So I don't think we've really seen that. But I think, I keep saying to everyone, you have to understand, we've only known about this virus, for what, 4 or 5 months. And we've been sort of trying to fight the fires here. We haven't had time to sort of go and sit down and write protocols and so on right now. But I think that some of the innovations that really are outstanding in our country so far have been understanding the phylogenetics of this virus. The first sequence that was produced by the NICD, there have been several more sequences since then. We have some idea of what the genetic clock looks like in this virus. We have a pretty good understanding of what its spike protein looks like. We have a reasonably good idea of the cytokine storm and how that leads to patients going quickly downhill, requiring ICU care. And we have, studies underway of those understanding pathogenesis. There are some studies underway looking at CPAP as an alternative, or not alternative, but at a different stage in patient care before ICU and mechanical ventilation. So those are some of the research ideas out there that are being done or being planned.
Okay, thank you for that. I think we have time for one more question, and I would like this one to be about the work that you're doing at Caprisa. And have you had any situations on your any non-COVID studies where the patients have tested positive during the study? And how have you handled that if that is the case?
Thankfully, not yet. Our clinic has been operating only on an emergency use basis. And we have put in place several procedures to mitigate risk. The big concern we have is an exposure. So a patient walking in that gets past our screening procedures, because we have a symptom check before a patient comes in, and if they get through those processes and come into our clinic and turn out to be positive for COVID later, then that means that everybody in the clinic who had an interaction with this patient has now been exposed. They all have to go home and sit at home for 14 days. So we have to be very careful how we function. So we've put all kinds of procedures in place to deal with that problem. One of which is our staff are on roster. So you don't have the same team coming to work every day. They are completely different teams running. There are some people that are common, which are not an issue because they don't have direct patient exposure. But our patient exposure people are on roster, so that if we do get an exposure, it doesn't mean everyone has to go home now and that we have to close the clinic, right? It's team B comes in at that point and they come and run the clinic. So we've got a whole set of procedures along those lines to deal with this problem. Now that the Department of Health has indicated that clinical trials can proceed, and especially for HIV and TB, where those trials are well underway, we can anticipate that over the next week or two, we will all start getting to a point where we are getting ready to start seeing patients again in our clinical trials. When that happens, I can assure you we're going to have COVID patients. It's going to happen. We're going to expose everyone in the clinic. And the key for me, don't panic. That's number one. Don't panic. Just sit back, calm down. You're expecting this. What's your procedure? What you're going to do? Right? And the worst of it all is when somebody amongst the staff turns out to be positive, and they have to start listing all the other staff that they worked with. Don't discriminate. Don't hassle about it. That person just got COVID. It's just what large numbers of us are going to get in this country. So just deal with it in a very calm, collected manner. And I think if you do that, you should be fine. I have to leave. I'm running a few minutes late, but I hope it was helpful and useful to all of you to get some sense of what the challenges are as we think about this epidemic, and how all of us in doing research and clinical trials need to find a way to do that in the midst of this epidemic. I wish you well and all the best. Thank you.
Thank you, Professor. Thank you very much. Enjoy the rest of your day. Okay. That was a very interesting discussion and presentation by Professor Salim Abdool Karim, and we are going to, just to remind you, compile the questions that you have posted and make those available with the responses from Prof. Abdool Karim once the presentation, once the webinar has closed. So in the we are now going to move over to the next item on our agenda, and I see the slides are already up. Thank you very much. And I would like to hand over to Becky. And Becky is one of the SACRA Executive Committee members, and she's also the regional trial manager in a pharmaceutical company. Thank you, Becky. We have your video there, and your presentation is up. So over to you.
Thank you. Thank you, Nikki, for
thanks, Becky.
Introducing me. You can hear me clearly?
Yes, we can now. Thanks.
Okay. I will be talking to you following information that we receive from different stakeholders that we work with in the clinical trials industry. This information comprises of how they have managed since the onset of the COVID-19 pandemic. So, firstly, just to break the ice, and after such a great talk from Prof, I just need to mention that today, as we commemorate International Clinical Trials Day, we celebrate the engine behind the frontline medical staff. These are the people working tirelessly to find solutions to the medical challenge that invade our communities. We celebrate the research participants who volunteer their bodies as incubators so that you and I can have a long life as we continue to do the right thing amidst all the challenges. So the background that I have, in our industry, we plan for everything. We have risk management plans. We have plans to manage the risk management plan itself. However, this pandemic, which has invaded the world, has shifted life as we know it. The greatest question we have is, how are we going to manage, and how are we managing? And it's good that Prof was with us this morning, and he has shed some light on how we're going to move going forward. However, it is important to share the experience and the lesson from the stakeholders within South Africa. Firstly, we comply to the National Disaster Management Act. We follow all the rules that are according to the lockdown restrictions. We also follow the local and the international regulations and guidelines. Within the local regulatory guidelines, we refer especially to the SACRA guidelines. We also follow local ethical committee guidelines. So each and every committee has published many different guidelines on how we should continue to manage during this time. And we also continue to consult with the ethics committee and also the local regulatory authority in an aspect that remains unclear to us. I just would like to highlight a little bit more from the SACRA policy on the conduct of clinical trials during the current COVID-19 pandemic. We need to ensure the safety of the trial participants and research staff. We need to maintain compliance with the current good clinical practice while trying to also minimizing the risk to clinical trial integrity. So we know that during this time, we are unable to meet protocol-specified procedure, such as administering and using investigational products, adhering to the protocol-mandated visit, laboratory, diagnostic, and many more of other activities that we need to comply to. SACRA does recognize that this protocol modification will happen during this time, and there may be unavoidable protocol deviation during this time. The measures need to be implemented to manage this time will vary also greatly dependent on many factors, including the nature of the study, the nature of the disease, the trial arm, the region in which the study is being conducted. So this is some of the information that we've gathered from the different stakeholders on how they have been managing. This is not necessarily the way in which everyone should go about, but every stakeholders within the clinical trials industry in South Africa, they are operating at a risk level, and they need to apply their different assessment based on the situation which is facing them on managing that particular protocol. We need to ensure that as we continue with essential medical activities, we follow all applicable regulations. In certain instances, halting of recruitment has also been advised. We also have noted that there are research sites that are located in highly impacted hospitals which are treating COVID-19 patients. Some of these hospitals are already considering finding alternative sites, finding secondary structures that they can use at this time to ensure that they continue with clinical trials activities. In some hospitals, they also had to consider moving clinical research activities to admin blocks, which helps because the clinical trial patients will not necessarily be in contact with the rest of patients. Additionally, we need to develop new processes to manage the studies. We need to prioritize continuity, performing risk-based assessment with a foresight that no one knows when this global challenge will end. These are some of the examples of the processes that have already been implemented at the different sites, which have noted that some of their studies had to continue based on the risk assessment. So at the different sites, site staff have been divided into staff that are admin staff and staff that are for clinical block. Number of site staff at a time is also limited. When patients arrive in a waiting area, social distancing has been implemented. This also is happening when it comes to bed setting where there's in-hospital patients. And these have been put apart from 1.5 meters to 2 meters. And they are also reducing the number of patients at the site at the same time, and streamlining the process to limit the time that the patient needs to spend at site. Additionally, we also comply to the different protocols, including hygiene practice, and then the patient screening for COVID-19. I will not talk much about this, but Prof has also reflected on that. But it is important to also go back to the SACRA regulation and also the latest regulation published by FDA, that there may be other activities that we need to implement, such as those that will require pre-screening for COVID-19 for patients that are in clinical trials. And in those documents, they clearly mention that we may not need a protocol amendment for that particular kind of testing. So it is very important that if we are continuing, even at this present moment, we do consider to start implementing processes to test our patients. And we need to ensure we continue following the tool guide that has been presented now and see what is applicable for the different sites. These are some of the activities that have been happening using still the risk approach by the different sites. Some of the sites have noted that on-site visit will continue for patients in their practices. This is based on a risk assessment. This is based on the kind of studies that they are actually conducting. However, obviously, the sites continue to prioritize patient safety and clinical staff safety. Many visits have been changed to phone visits. The patients that need to come to sites, they are being transported differently rather than come through using public transport. And at certain points, visits are happening at home and visual visits are also happening. Some important aspects to consider is to look at your informed consent and ensure that whatever activities that you are implementing are already covered on the informed consent. And if they are not covered, it is important to start implementing processes that will address information such as home visit, IP delivery, change in visit schedule, and also having alternative site address. So these are the kind of things that we need to start implementing in our processes. Safety monitoring, measures to continue monitoring of patient safety needs to be put in place, and obviously measures on how safety data will be collected while the patients are not coming to the site. Additionally, some of the processes that were highlighted: clinical supply management from site to patient services, direct to patient services, ensure that consultation is done with the relevant authority when this kind of services are implemented. On-site collection by family members for particular subjects that are at high risk if they are to not comply with social distancing. So you can actually start requesting family members, for example, to collect the clinical supply where possible. Laboratory assessment, where it is advisable by the sponsor and also in consultation with a particular central laboratory, we can implement local lab for safety testing. And for primary endpoints, there is other means where we can retain samples frozen for longer than 30 days. This can happen only after consultation with the different parties, but some studies have actually implemented this kind of processes. Some other aspect that we still need to consider is how do we continue with quality control and quality assurance at the different sites. So some of this information are just still draft. Some of this information is still what we need to implement. When we come to monitoring, on-site monitoring has been halted at many sites in South Africa. If we are to resume monitoring based on the information that has just been shared now by Prof, how do we ensure that we comply? And it will not possibly be at all sites that we will be able to continue with on-site monitoring. The latest guidelines does highlight that we can optimize remote monitoring activities. With consultation with the sponsors, we need to identify how can we implement different kind of strategy when it comes to remote monitoring activities. The FDA guideline just highlights that they do not necessarily have a specific on what remote monitoring activities is. So it is important that we consult, obviously, with the different quality bodies within our organizations to see what kind of monitoring can be implemented. Data management becomes very important at this point because it's mostly about tracking. It's mostly about tracking of the data that we are not able to collect. How are we collecting that data? How are we making that particular data available so that we can be able to identify and put together the records that are needed to report the protocol deviations, for example. So it is important to work together to ensure that we are also continuing with the aspect of data management, also depending on the different studies. SACRA again recommends an approach to the new processes. Those are some of the content that we, as the stakeholders that have put together that information, thought it's critical to have as part of your process. And SACRA does recognize that a change in policy and procedure will be needed to obviously address issues that are impacted, but not obviously limited to the informed consent, the study visit procedure, data collection, study monitoring, how we report adverse events, the changes at the site when it comes to the site staff, and how we are going to travel. So it is important to ensure that your process that you implement does cover all these different aspects. This is an experience that we have shared from one of our contributing stakeholders. This is the direct to patient. This is very critical to also note that we are ready in South Africa to implement services such as this. COVID-19 gave us an opportunity to actually realize how flexible we can be, and we are able to implement those kind of services. However, everything needs to be done in accordance to regulations. Currently, we ensure that when we are transporting, all the people that are working are equipped with the PPE, and they are avoiding contact with the product and the patient, and they apply social distancing. The staff are also trained, the courier companies are trained, the temperature monitoring is also ensured as the drug is traveled through the different areas. We also need to ensure that we have documented patient agreement. We also have acknowledgement of receipt of the particular drug, and we also need to have records of on-transit temperature monitoring. Patient agreement in this time can come in different formats. Some protocol already have those kind of direct to patient services included, and some informed consent could possibly already have those kind of services included. If they are not included, it is important to ensure that the patients have given authorization for those kind of services, and your ethics committees and your regulatory authorities does authorize that. So this is the future of clinical trials. COVID-19 has given us an opportunity to look at ourselves and say, as South Africa, where are we when it comes to this, some of these different aspects that were reflected by the stakeholders? Where are we when it comes to conducting virtual trials, in-home study procedures? What regulations do we need to look at when we look at our SAGCP, for example, when we look at our ethics committee's guidelines? At-home nursing services, what services do we need to start implementing? What are the areas that we can actually reach out to, looking at how our sites are mapped? Where can we actually start implementing those kind of activities? Because we will need this. Telemedicine is here and it's being used. This is our time to also explore how this can be best implemented in clinical trials. Electronic data collection. We have a lot of experience when it comes to electronic PROs because we usually use those for diaries. But it is important to realize that we are ready for this. We have patients that are already using this kind of services. So those are the kind of things that COVID-19 has opened our eyes to. Electronic trial master file, so many organizations have started and some have already implemented. But at site level, how do we move to a non-paper-based way of working? Implementing of e-consent, where are we as South Africa when it comes to our regulatory standards and our ethical requirements? How do we comply to the requirement of implementing electronic consent? Because the patients may not be able to come to the site. How are we going to be able to collect those kind of documentation? Use of electronic source documents. How ready are we if we look at our site infrastructure? Is our site infrastructure compliant? Can it be qualified as a system that can produce electronic source documents? The use of electronic health records. This becomes critical, especially where we need to recruit patients from public practice and we need to have access to medical records, for example. How are we going to access medical records currently when we know that the paper also transmits COVID-19? That is another way of where we should consider to think, how are we going to move the paper all around in our way of working? The local laboratories, the role of local laboratories in clinical trials. How do we start working together with our global partners to ensure that local laboratories are equipped to actually provide the needed for clinical trials? Our site guidelines, our site SOPs, we need to adjust them. Obviously, at the sponsor level, we are also working together on this because this was unprecedented for all different stakeholders. The site's responsibility in managing trials. This is what we've seen during this time. There is more expectation from different sides. The sites have to put in the work. The risk is basically primarily at site, if we think about it, on the transmission of COVID. So how do we look at our responsibility at the different sites? What systems do we implement, as Prof has already highlighted? What is it that will need to be put in place to actually ensure that we continue with clinical trials over this time? I know I'm not providing answers, but I'm just providing this information that has been provided by different stakeholders, which is basically at this moment primarily questions, and we are all trying to work together to find solutions towards that. There are opportunities for South Africa for the future of clinical trials, the ethics on conduct of clinical trials, our regulatory response to the future normal, the clinical trial budget, the use of local laboratories, the implementation of home nursing services, and the timelines in the way we respond and in the way that we actually are active and ensure that we are ready to implement different kind of trials at this particular time. How do we move along with the global trends? So this is also very important, and I need to mention, because we cannot continue and think that all people are not affected differently when it comes to this time. So there is an impact on income reduction. Some sites, some sponsors, some laboratories, so many other aspects within our stakeholders have lost income. And obviously, the government has published different ways in which we can apply for the different funding or different support that is needed. Some has been caused by delayed or termination of upcoming trials, and then some have been caused by less activities on site. So all these different aspects does affect how our future will look like. So we need to actually start looking at collaborating even more in order to equip each other. And I would like to thank these contributors, Dr. E. Mita, who is the principal investigator at Newtown Clinical Trials, Dr. Dr. Tayop, I usually call you Dr. Tayop, who is the MD at Medinova, Rob van der Berg, the director at Pharmalogic, Prof. Burgess, who's the principal investigator at Trade Research, Ms. Catherine Land, a director at OnQ Clinical Research, Janet Greb from Vet Shanduka Research Center, Dr. Michelle Middle from PharmOps, and Dr. Sanet Aspinall. Thank you so much for contributing to this presentation. Thank you, Nikki.
Thank you very much, Vicky. Thank you. I appreciate the presentation. It was really enlightening, and thanks to all those who did contribute to the development of that presentation. Again, if you have any questions that you would like to pose to Vicky, please do so in the chat panel, and we will get those to Vicky. I'm sure she'll be happy to take a look and to respond to anything that you have to ask, or any information that you would like to share with her. I'm now going to hand over to Len. Len, just to confirm that you can hear me before I... Okay, we are seeing your screen, and yes. So Len, also known as Leonard Lazarus, is the African Regional Director of BioCare. He is also the Treasurer and serves on the SACRA Executive Committee. So over to you. Thank you, Len.
Thank you, Nikki. Can you hear me?
Yes, we can. Thanks.
All right. I'm not using a webcam. I have my webcam off, so everyone knows. I'm just going to share my slides. You can see my screen?
Yes, we can. Thanks, Len. You're good to go.
All right. Just an introduction to BioCare. So BioCare is the global logistics courier provider for over 30 years of dedicated experience in the pharmaceutical and biotechnology and life sciences sector. Specifically, we provide dedicated specialized logistics services, both systems and people, for the scientific sector, and our services are most comprehensive of its kind available on the market. On the screen, you'll see some data that I've shared with you of some KPIs and shipments experience. Okay, the company was built up a unique client-centric approach by employing frontline scientists in frontline logistics positions and assembling a team of best-in-class industry experts in quality, cold chain, and regulatory compliance, amongst others. We focus on providing the most comprehensive, time-sensitive, and temperature-controlled logistics available, whilst delivering flexible, tailored, cost-effective solutions to all our clients. Our network expands over 160 countries across Europe, Africa, Asia, and Americas. Some of our portfolio of services we cover. We cover aspect of shipping process, providing you with the comprehensive and convenient global logistics solutions. Some highlights is the global logistics service, regulatory compliance, temperature control solutions, customer-focused solutions, and value-added services. The global logistics services expands across our network to GDP standards and ISO 9001 accreditation. Our local offices provide expert knowledge of regional, national, and international logistics. Our compliance team have dedicated considerable time and resources to ensuring our global logistics solutions fully comply in all elements of import and export regulations, whilst we offer time-sensitive saving service, assisting you with the assignment of customs tariff codes, classifications of dangerous goods, advising you on your legal obligations before transporting your consignment. The temperature control solutions have an unbroken temperature chain could not be more important both to the success of your product and to your patient's safety. From the moment your shipment leaves your premises to the moment it arrives with its end user, you need to be certain that it's in the hands of the experts who can maintain the correct temperature at all times. BioCare has a comprehensive set of contingency plans in place that will ensure coolants and replenishments along the way are moved in the appropriate temperature control storage facility should there be a delay in transit. This will obviously guarantee you that the consignment arrives at its final destination in perfect condition without any deviations or damage incurred along the way. In saying that, in line with COVID-19, there's a lot of regulations that goes into that as well, into the regulatory compliance. From a South African perspective, it's governed by ITEC, together with Department of Transport and Industry, and certain tariff codes, especially on your investigational products and time-sensitive material that's exported out for studies within the African region, is actually required to have an ITEC approval prior to shipping. This is one of the regulations that came into effect from the beginning of lockdown. We went into lockdown in March. That has slightly been relaxed as we move down the levels. In level 4, the regulatory authorities do not specifically need your CIPC certificate anymore, just the approval from ITEC. And we'll see that change as we move down the levels into level 3, as Prof was saying on clinical trials, should be uplifted in the next 2 weeks. On the customer-focused solutions, in recent times, and surprisingly enough during lockdown, a number of logistics companies, forwarders, specialist couriers have been increasingly inflexible when it comes to meeting their clients' requirements. Our team at BioCare are proud to be dedicated to bucking the trend, doing everything that we can to tailor our services to our clients' specific needs, however challenging or complex it became and becomes in the future. And part of that is obviously having our teams around the clock with time differences, working on different projects to make sure that the client needs are always met. The value-added services, we are extremely proud of the BioCare difference. Our focus is supplying highly comprehensive service options while also delivering the most tailored, flexible, and cost-effective global logistics solution that meet the needs of every client. To this end, we are proud to provide a wide range of value-added services that ensures our clients can benefit even more from the specialist solutions that we provide, including consultancy and real on-time monitoring. Some of the flexible solutions, I've put some case studies up that we've done since we went into lockdown. We've handled and transported investigational medicines, product and samples during all phases of clinical trials, irrespective of temperature, irrespective of classification of infectious, non-infectious. We've also completed all the necessary paperwork we're legally required to, allowing consignment notes, airway bills, and custom declarations and certificates by assisting the clients for transport. And on this trial, you see BioCare provides specialist management of clinical trial supply chain, and we can be involved as you need us to be, from setting up the complete logistics of your study, liaising with the different collection and delivery sites, to delivering single shipments within a large trial. We can complete all necessary paperwork as legally owned. The image that you see on the screen is one of the first PPE shipments brought in from China that came in on the 737 that we brought in for, and that was delivered to principal investigator Dr. Nigel Garrett and study coordinator Bongi, that's not in the picture. The highlight was delivering this PPE to site, which was really exciting to see the enthusiastic faces when arriving at the site. This was delivered by our regional manager in Durban, you see on the picture, Colin Pillay. During COVID-19, we had to do a lot of flexible logistics solutions. Our recent customer custom solutions included some temperature-controlled plasmids for South Africa. This was in the very early stages. This is for Dr. Alex Siegel, who actually works with Prof at Capricum. Dr. Siegel is a member of the faculty of the African Health Research Institute and a research group leader at the Max Planck Institute of Infection Biology in Berlin. The request came in from Dr. Siegel for plasmids and his team for developing COVID-19 research in South Africa. And I'm quoting a message that Dr. Siegel sent to me. He says, these reagents that BioCare delivered to us from the US at a record speed and surmounting considerable obstacles because of the lockdown, both in the US and South Africa, will allow us to understand the immune response of people in case they're into the virus. The shipment contained the human gene fragment which allows SARS-CoV-2, the virus causing COVID-19, to affect human cells. It also contained a set of gene fragments allowing us to make non-infectious SARS-CoV-2 that is fluorescent. None of these reagents are dangerous or infectious. In combination, these reagents will allow us to test how quickly people in South Africa, including those with HIV infection, are able to fight SARS-CoV-2 infection by producing antibodies that stop the virus from infecting the new cells. So that's one case study. The second case study involves PPE that actually had to go into Germany, Italy, and the Balkans, Albania. Some of the locations that we had challenges that we faced was collecting from Shanghai to ship to these countries. And the export regulations in those places, poor availability of cargo space out of China, complicated customs clearance process. Some of the solutions our teams put to globally was a dedicated cargo solution to Germany and Italy, an hybrid cargo solution with warehouse and service partners. The results for that was the bespoke service, quick transit times, and a 360-degree service. The case study 3 involved leading hospitals in New York, which we've all seen on TV, with the high shortage of N95 masks, which we had to import from China. This involved challenges moving 11 pallets without flights in place with limited space, became very challenging. We had to ensure all the paperwork is correct in place prior to flight departure, and with both local and US regulations and tax exemptions in place with flawless communication. The solution that was provided was the shipment needed to be picked up immediately and stored in the warehouse until all export documents received approval. We worked with the export broker both in the US and in China to actually expedite the process, and we were able to secure flights shortly after that to the US. The results for that hospital was they received the 200 masks for their staff to stay protected on the front line. Some of the other case studies that we've done, the picture you see on your screen is of the British Army, who set up specific warehouses to deal with COVID-19 and suppliers. And BioCare was actually involved in the logistics. And obviously, we're not allowed into these hybrid warehouses. And we had the Army actually working within these facilities to actually bring the stuff out to us. Some of the other challenges that we've done during COVID-19 was investigational product into Liberia, which was needed urgently for patients. And in that case, we actually had to HR an aircraft directly into Liberia. And obviously, you know, with the unrest in Liberia, it was a very quick turnaround. We obviously, the client and the Army met us on arrival. A plane was turned around, I think, in 10 minutes after arrival and back out to departure again. The South African team worked very closely with COVID-19 from the beginning. We were involved in the first COVID samples out of Botswana for the Ministry of Health in Botswana, driving regularly on a regular basis through Botswana and back, bringing it back to the National Institute of Communicable Diseases. And that happened for the first 6 weeks. And thereafter, Botswana, we assisted them to be able to actually run their own testing in-house in country. Some regulations outside the BioCare presentation, that as Prof mentioned, is COVID-19 is something that's going to be with us for months and years to come, and some of the restrictions on regulations from customs and the regulatory authorities in South Africa is a lot of stuff is restricted coming in, especially your PPE, ventilators, thermometers, etc. And majority, or about 90% of this comes in from China. So the restrictions have been placed on both sides, in China and in South Africa. And in order to ensure that you have a smooth logistics solution to carry on as we move into clinical trials, PPE will become more important for sites and site staff. Is this PPE needs to be in place. And one of the regulations that needs to be followed, this will be controlled by the Department of Trade and Industry, DTI, and ITEC, and it'll be given on a per shipment basis. A permit will be issued for 6 months. So, and those are some of the regulatory expertise we've actually had to adopt in-house to actually ensure that the transition goes very smooth with ITEC. And I think that is it from my side, Nikki. So just the network, that's our network of offices in the 160 countries.
Thank you, Len. That was very informative. I would like to hand over, I hadn't shared that. I would like to finally hand over to Teresa Skanes, who's also on our SACRA Executive Committee, and Teresa is the Vice Chair, and Teresa will be closing today's call. Thank you very much. Are you there with us, Teresa?
Hey, Nikki. Yes. Hi, everybody. And thank you very, very much for what I believe has been a phenomenal event. I've learned so much from this whole experience, and just to put a few things in perspective. So I think at this time of new experiences in our clinical trial setting, we're learning every day. Today being International Clinical Trials Day, I think the need for us to relentlessly pursue new and better ways of doing things is top of mind. We've had some phenomenal insights into the current situation and some ideas on how to proceed under this new normal. So SACRA really hopes that each and every one of you will take what you've learned with you today into your communities, both here in South Africa and indeed internationally. And remember, don't panic. I think that was one of the big things Prof taught us today. Don't panic. Check your COVID toolbox and put appropriate measures in place. Get in touch with your ethics committees and make sure to continue correctly to do what needs to be done while we learn to live with this virus. So I want to say thank you to Professor Abdool Karim and his team at Caprisa. Thanks for their support for SACRA. Thank you to BioCare for your support. Thanks to the entire EXCO who put this together, and for the local and international participants who took time out of their day to join us in South Africa for this informative session. There are 2 additional pre-recorded sessions that are available on SACRA's website after this. And this particular session, this webinar, will also be made available to everybody that registered for this event today. So check your emails. There'll be a feedback survey available, and we hope that you'll all provide some feedback to us so that we can build on what we have done today. So I wish you all a happy Clinical Trials Day, and I hope that you all are really proud of what we do and how we do it, and that we continue to provide a good service to our communities. Thank you.